Welcome to Science Frontiers, a space for discussions in the frontier areas of science and technology. I am your host, Binay Panda. Synthetically produced organisms are often called the living machines. What are those, and what are the differences and similarities between actual living organisms and living machines? In the second episode of the series on synthetic biology, we shall discuss about the differences and similarities between biology and engineering. This is especially useful for students who are still undecided between the two subjects and where one must choose one over the other. We shall continue our discussions with today's guest, Pawan Dhar. Pawan is a professor in the School of Biotechnology, Jawaharlal Nehru University, New Delhi. Pawan, welcome to the show and thank you for joining us.
Thank you, Vinay.
The first question is, there's so much of talk about the applications of engineering principles in biology. Can you tell us a little bit about what are the differences between engineering and biology?
Wonderful. Thank you so much, Vinay, for hosting this episode on finding parallels between engineering and biology, because a lot of discussion has already happened about how much does an engineering fit in, and does it fit in at all? What are the good case scenarios and boundary conditions? So I think it's very important to draw similarities and differences between these two fields. So let me come back to the question, what are the dissimilarities between engineering and biology? You see, when we make engineering components, for example, a computer that we are interacting with, the components are essentially tagged or latched or anchored to a substrate. It could be motherboard, it could be some tube, it could be some wire, something they have to hold fast to. But in biology, as you know, DNA, RNA, protein, they're all swimming out there. It is not held onto something, it's only constrained by some physical space. So that's a fundamental difference, because these molecules can go from any point to any point within that space. As against engineering system, when the information transfer is hardwired. And the second difference between two is that the design principles in terms of the rules, laws, models are pretty much established in engineering. While in biology, all we have is laws of, if you call them as laws of inheritance, or you may call them as ratios, that's also fine. We have Hardy-Weinberg equilibrium, or we have metabolic scaling laws. But these are all the correlations or regularities one observes. These are not useful when you're constructing something. Because for construction, you need standards and you need rules of composition. And these, all the three categories that we have just gone through, they do not supply you that information. They were made for something else, which is fine. Third point is biology is analog, not digital. When you have engineering technologies based on engineering practices and principles, these are digital systems predominantly. But biology is analog, as you see in case of fan regulator, you go from 0, 1 to 3, 4, 5. This is an example of a very, I should say, approximate example of an analog system. It may not be a perfect example, but it is just very close to that. Last of all, in biology, you have the phenomenon of emergence that you don't find in the engineering systems. Emergence means that when you take components by themselves, they behave in a certain way. But when you combine the components, they behave in a different way. This is unexpected behavior, as you find, for example, when there's a school of fish. The way they move, you take an individual fish, you can never figure out how it will move in that space, the water. Or you see a flock of birds, the way they move, they cannot be predicted simply by looking at the bird itself. When hundreds of them come together, they show certain patterns which could not have been imagined by looking at the bird. So these are the dissimilarities between engineering and biology, and that's why it makes very interesting to apply engineering concepts to biology.
Thank you, Pawan. And I think you mentioned three things, and we'll touch upon those in detail, some today and some in future episodes. First of all, you gave a very interesting analogy between the motherboard, the computer motherboard, and the biological system. And the second, what you also said, the differences between the the analog and the digital system. And the third, the sum of all parts may not be the one that you can deduce by looking at individual parts trajectory. So now that we understand the differences between the discipline of engineering and biology, and given that engineering approach has found expression in the form of synthetic biology, can you talk to us about the similarities between those two disciplines?
Yes, Binay, definitely. So now what are the similarities and what is the use case of engineering and biology, and why engineering communities started the whole idea of synthetic biology is because both the fields, both the disciplines are based on robustness. So what you construct is robust, whether it is a bridge or a building or an organism. They're robust systems. They're based on linear and non-linearity. And the components that make them, they show multitasking frequently. So for example, if I take robustness as an example, then let's say if there's a building and there are certain buildings in earthquake-prone areas, as you have lived in, Vinay, and you know that when there is an earthquake, the force is distributed. It is not, well, all the buildings are designed for a vertical tolerance of the pressure, the weight and the gravity. But when there is a horizontal force emitted from earthquakes, then it is that there are certain buildings that are designed to diffuse the force, thereby showing robustness. Likewise, when you look at the organisms, you knock out a gene, you may not find anything happening there. It may be happily dividing. You may knock out 2 genes, 3 genes, unless you knock out a certain combination that is critical to the functioning of the cell. It may even forget or it may even ignore. Not only that, people have even gone to the extent of removing some of the major blocks in E. coli. And they found that these were the evolutionary junks. And cell was able to withstand it and happily divides. So robustness to me is one big similarity between two systems. Linear and nonlinear processes happen in both the systems, and multitasking components. On an average, it is expected that a protein will interact with 5 different proteins on an average. So likewise, you have the same multitasking ability in the engineering systems.
Excellent. Let's just go back for a minute to the plug and play system that you mentioned with an analogy of the motherboard. So, when we were young in the college, we used to build these motherboards from different parts and build the desktop computers, just buying different parts and assembling it. So can we think like the non-living system, like the computer, can we do a plug and play kind of a situation in a biological system? Can certain things be replaced? For example, if you compare a computer with an individual cell has multiple different parts, multiple different organelles. Now, let's say, for example, it has a mitochondria, and in the plant cell, there are chloroplasts, there is a nuclear DNA, there are different organelles. So is there any example that have already been set or people are working on where a plug and play situation may work? That means you replace one organelle or a part of a cell from another one, and it still performs very good, or it actually acquires new or additional characteristics from where the organelle or the part of the function has been borrowed from.
Wonderful, Binay. I think that's a very important point because what you're now hinting and slowly taking the audience towards is editing and writing. So when we go towards the editing part, when let's say there's a system which needs a certain plugin, can we do a biological plugin like running an app? Can we run an app in a system? Yes, it has been already tried. Back in 2000, year 2000, Michael Elowitz and Stan Leibler, they published this paper on repressilator. It was published in Nature magazine. And they showed for the first time that a 3-gene circuit can be plugged in, and it shows a certain predictable behavior over many generations. So the fluctuations or the wave-like oscillations of the gene expression was visible. So this was the first example. Of course, after that, people had tried many combinations. And the extreme form of this, as you know, was when Craig Venter replaced the original genome with a new genome. This is a large-scale edit of the cell, and his team was able to demonstrate for the first time that you can boot up a cell with a brand new genome. So you keep most of the hardware intact, just replace the software, and still it will function. Also, we have seen this project from John Hopkins group. Chandra has led this. So now they published a paper in Science several years ago, and they demonstrated that one can chemically synthesize a chromosome. So you take the sequence of the chromosome, you remodel the chromosome, make it lighter, and then chemically synthesize the entire chromosome. So it took 5 years for the person who was actually on the job, on the bench, and reboot the cell with a brand new chromosome, and it was working. So these edits depend on your, of course, the funding, also on your competence. But right from small edits to big edits, people have tried different things. They have tried to create logic gates. They have tried to create apps. But then when you talk of gates and apps in biology, it's not exactly the way it happens in engineering, because these apps and gates, they lose their property over time.
Excellent.
And that's a big problem.
This is very interesting. And I just, for our audience who may not be aware, I want to say that this is very pertinent, because when Pawan, you talked about what has been achieved for the synthetically making chromosomes, and in fact the project which is called the East chromosome in making, so East 1.0, East 2.0, where people actually are making or building, rather, in engineering terms, the entire chromosomes, have been successful. And this is very pertinent to talk about it today, because even 50, 60 years back, the legendary work of our own Hargobind Khurana and Marshall Nirenberg, actually, who got Nobel Prize, and the whole process of starting to make synthetic genes now yielding fruits in terms of building the whole new organisms. And in fact, this is also very apt to remember Dr. Khurana, because he recently died. And in fact, we should celebrate the legendary work of Dr. Khurana and many others who have actually paved the way of this new field of synthetic biology. We will be talking about the synthetic genomics approach that you mentioned, Pawan, in our future episodes, about how people are trying to build the whole new cell. And you mentioned about Craig Venter's work, and of course, that is very foundational, and we'll discuss more in the detail. Now, coming back to the fundamental question that where we started this episode, for example, there is a student who is undecided whether to take engineering and biology. And as you know, in our country, the walls between these disciplines are very thick and very long. So you are thinking about engineering in the terms of taking whether computer science, electrical engineering, mechanical engineering, aerospace engineering, civil engineering, and biology doesn't really fit in. So I think this is one of the things that the students might want to know. If someone takes biology, as you mentioned very eloquently during the first half of this talk, is that you still can use the principles of engineering. You can still use the principles of building, although biological systems or the living machines are foundationally and fundamentally different, but the way it has been built is also very similar to that of the non-living organisms. So do you have any word of advice for students who are undecided at the moment whether to take engineering or biology, and how someone can go about choosing a subject over others? Because as you know, unfortunately, in our country, we don't provide the flexibility of choosing both at the same time. Although I must emphasize, with the new educational policy 2020, we do have some emphasis of getting credit, or the academic bank of credits, where one can do mix and match, but it's yet to be practiced. So do you have any advice for our students?
Thank you, Vinay. I think that's wonderful, because we keep hearing this over and over again. How do the two disciplines fit in, and what is there for an engineering student? Well, by default, engineering students have very little knowledge of biology. They're not supposed to know, because they have a different discipline in mind when they join engineering. However, in my view, the easiest plugin for their thought process and their training would be if they focus on building standards in biology, if they focus on building IEEE equivalent standards in biology, because then they will connect with what is happening in biology, because there are no standards. So the foundational work has yet not been completed. It might take another 2 decades or 3 decades to complete the foundational work. There's a lot of scope for engineering students who understand IEEE equivalent standards or IEEE standards, without which they cannot build anything. Now, can they transport those concepts to biology? And can they tell us what are the different ways of building the standards? Is it a community-driven, is it an institution-driven, or is it like a ground-up engineering, building of standards? Or because there is a lot of variation in biology. Let's say if you want to do cell culture, and in your lab and my lab or in another lab, we will probably use 3 different methods of cell culture. Or if you have to purify protein, we'll use different methods. And in all the labs, these methods work. That's why we're using them again and again. Now, this needs to be somewhere. When you look at the the standards, building the standards around variations in biological processes, when you look at those needs, because different labs have different ways of doing the same thing. So now there's a need to build a standard registry of processes, which means that this registry would essentially say that one has to use a certain process because there's no standard process. And once you are done with the process standardization, then you come to the component standardization. And then you build the standards, and building the standards, well, there are many aspects to that. I will not be able to touch all of that today, but probably for an engineering students, it would be better if they focus on building IEEE equivalent standards in biology. That's now number one. And number two, there is no, right now, there is no unit of current in biological circuits. If you look for the unit of current in biological circuits, there is no such unit. And that, to me, is going to be excellent from engineering perspective, because they understand, the electrical engineers understand what is in unit. And to find a similar unit in biological circuits would be intellectually fascinating.
Thank you, Pawan. In fact, you have given us a hint and clue what our next episode is going to be, which is on biological standards. So due to time, we are going to stop here for today's episode. But for our viewers, I want to tell you that we have very exciting new topics within the series of synthetic biology and many more interesting topics coming up. So stay tuned. Thank you once again. And thank you, Pawan, for being here. Thank you.
Thank you. Thank you. Thank you very much. Have a good day.